Plants plays a major role in our lives. They are the primary source of our food, which help us to maintain our health and well-being. Many plants are also used in various ways, such as medicines, trades, and for aesthetic and leisure purposes. With these benefits that we get from plants, have you realized how and what will be our future without them? What if one day all plants go extinct? How could we prevent such circumstances? Technologies have been developed and implemented to preserve planting materials that will serve as a germplasm reservoir. One prominent method is the infamous cryopreservation. So what is cryopreservation all about? In this lecture video, I will briefly discuss cryopreservation, its importance, and its different techniques. Cryopreservation was derived from the Greek word cryos, which means cold. So, from that basic definition, we can say that cryopreservation is a practice which utilizes low temperature as means of preserving seeds and plants. For a formal definition, we have here that cryopreservation is a process of storing or preserving live cells, tissue, and other biological materials in a deep freeze at sub-zero temperatures. Typically, the sample is stored at -96 degrees Celsius. So, why do we have to perform or do cryopreservation? So first, it provides great hope for the preservation of endangered species as well as indigenous plants. Ti endangered species nga makunkuna ket dijay mula nga dandani maibusen. Ken ti indigenous plants nga makunkuna met ket dijay heritage plants or mula idi agkakauna nga panawen. Second, it provides long-term preservation. For example, no agkaron iti calamities or natural disasters, at dalata iti pagalaan tayo iti samples or seeds nga mabalin manen nga imula. Third, for breeding purposes. Syempre no agde-develop da iti baro nga variety or seeds of crops, they will trace back the characteristics which will serve as their references. So based from the article written by Tolga Isgu and Yesim Yalan Mendy, there are different combinations of the cryopreservation techniques. We have here the A. Controlled rate cooling or two-step freezing, controlled rate freezing or slow cooling. B. Vitrification with plant vitrification solution -2 PVS2. C. Encapsulation or vitrification. E. Encapsulation or dehydration. F. Droplet method. G. Desiccation or laminal flow cabinet or silica gel. And H. V-cryoplate and decryoplate procedures. Slow cooling is given by a gadget named programmable freezer. In this system, the boundaries can be changed effectively like beginning temperature, freezing rate, last temperature, and holding up time. Since this strategy is costly, lower innovation can be utilized instead of. Vitrification is very effective strategy for cryopreservation of plant materials since it would keep away from the possibly weakening impacts of intracellular and extracellular freezing. This strategy can be characterized as the change of water straightforwardly from the fluid stage into a shapeless stage while staying away from the development of translucent ice. Vitrification is ordinarily utilized for embryogenic callus lines, meristems, physical incipients organisms, and so on in liquid nitrogen. Next, encapsulation or vitrification. In this method, the explants are encapsulated in alginate beads, loaded with vitrifying solution and dehydrated before being rapidly immersed in liquid nitrogen. Encapsulation or dehydration. So this method was produced for in vitro pear and potato auxiliary shoot tips. This technique depends on the conservation of engineered seeds. Next, the droplet method. The drop technique with PBS2 or DMSO is likewise utilized for cryopreservation of a few plant types. Drop of various grouping of DMSO or PBS2 arrangement are put on aluminum foil strips and drenched into liquid nitrogen. Although the cooling rate is incredibly quick in droplet method, no vitrification is settled inside the plant tissues or cells. Next, the desiccation method. This method, either in laminar flow cabinet or silica, is utilized for cryopreservation of substantial undeveloped organisms, engineered seeds, or early-stage tomahawks. Since this strategy is basic and simple taking care of it, it ought to be attempted first prior to testing different methods. Lastly, the V-cryoplate and D-cryoplate procedures. So this strategy is another cryopreservation techniques in view of vitrification and air parchness of explants put on aluminum cryo plates, named the V-cryoplate and D-cryoplate strategy. This technique is an easy-to-use methodology, and higher cooling and warming paces of treated explants are conceivable by straightforwardly submersion in liquid nitrogen. So in this age where biodiversity and decreasing space of wild areas is very alarming, the practice of seed preservation, such as the seed cryopreservation, is critically important to maintain the availability of genetic sources of both heritage and endangered species of plants. Preserving the seeds of various crops at present secures the future of next generations. That's all. Thank you.